Hello everyone, my name is Paula and today I'll be discussing the frontline role of Reg3 gamma in protecting small intestine immune homeostasis. We all know that intestines are packed with trillions of microbes, but this excess contact between the microbes and the epithelium causes overstimulation of immune responses. This can lead to inflammatory bowel diseases, also known as IBDs. So, how does the small intestine maintain a safe distance? Well, the study focuses on Reg3 gamma, which is a C-type lectin antimicrobial protein, and it is triggered by the MyD88 dependent protein signaling system within the epithelial cells of the small intestine. Reg3 gamma is what keeps microbes at bay from the epithelium. So, before this work, there were a lot of questions. Was MyD88 sufficient or even necessary to maintain this gap? What specific role did Reg3 gamma play? Did it kill the bacteria? Did it reshape the microbiome? Or did it enforce the gap between the microbes and the epithelium? In this paper, the authors were able to show exactly how Reg3 gamma functions and how it acts with the MyD88 to keep gram-positive bacteria away from the epithelium. So there are four main findings of this paper. The first one was that MyD88 was seen to maintain a 50 micrometer gap between the microbes and the epithelium. The authors used fluorescence in situ hybridization, also known as FISH, to measure how close the bacteria really got to the villus tips of the epithelium. They used red fluorescent RNA probes in the bacteria and blue DNA fluorescent DAPI in the epithelial cells. So how Reg3 gamma works is it binds to the peptidoglycan of the gram-positive bacteria and it accumulates near the epithelium, which creates a zone that keeps the bacteria separated. In this experiment, they saw that in wild-type mice, the bacteria stayed 50 micrometers away. However, in MyD88 knockout mice, they saw that there was no gap and the microbes just swarmed towards the epithelium. This shows that MyD88 signaling is necessary for maintaining a gap. The next experiment actually showed that MyD88 drives Reg3 gamma production in these epithelial cells. So they selectively deleted MyD88 from mice, and they saw that mice with this knockout had no separation between the microbes and the epithelium. However, when they restored MyD88, they also rescued the gap. Using immunofluorescence, they confirmed that Reg3 gamma levels actually dropped when MyD88 was knocked out. This showed that MyD88 actually acts within the epithelial cells to trigger Reg3 gamma production. The third main finding actually showed that Reg3 gamma keeps gram-positive bacteria away from the epithelium. So they looked closely and they found that species like Eubacterium rectale and segmented filamus bacteria, which are gram-positive bacteria, were right up against the epithelium when Reg3 gamma was knocked out. However, in the wild type, they stayed away from the epithelium and maintained a 50 micrometer gap. The last main finding showed that a loss of Reg3 gamma actually increased the immune activation, which we expected. So Reg3 gamma deficient mice actually showed fecal IgA levels that are 5 times higher than normal and an increase in IFN gamma producing CD4 positive T cells, which both show heightened immune activation. Then treating these cells with antibiotics were able to eliminate this effect, showing that these results were actually microbiota dependent like we expected. Here we have a general mechanism overview. In the first panel, you see epithelial cells detecting nearby microbes using pattern recognition receptors. In the second panel, you see MyD88 being activated within these epithelial cells. In the third panel, you see MyD88 driving Reg3 gamma protein production and secretion outside of the cell. In the fourth panel, you see Reg3 gamma binding to these gram-positive bacteria and keeping them 50 micrometers away from the epithelium. Finally, in the fifth panel, you see a balanced immunity outcome with no unnecessary inflammation. So, what does this all mean? Well, traditionally we think of gut immunity as maybe killing bacteria or killing microbes, but this study kind of just flips that around. Maybe we don't have to kill anything, but we can just have it maintained at a safe distance. This is important because this means that beneficial bacteria can actually stay around as well. Of course, there are a lot of questions that remain, such as are there spatial regulation systems for gram-negative bacteria? How do diet and maybe microbiome changes affect Reg3 gamma levels? And what really keeps Reg3 gamma from sticking in our small intestines and not just being blown away? We could use inflammatory bowel disease models to see if delivering the Reg3 gamma protein can maybe restore the gap. But at the end of the day, this shows that the small intestine immunity isn't all about just fighting microbes or killing them, it's about managing a type of relationship. And the information from this study can be used in the future to see if we can manage new ways of dealing with gut inflammation and other diseases. Thank you.